Viral genomes. Viral genomes can be in the form of single-stranded RNA, double-stranded RNA. It can be in the form of single-stranded DNA and double-stranded DNA. Viral genome, which is also called as viral chromosome, can be in the form of linear DNA and it can be in the form of circular DNA. A viral genome is the genetic material of the virus. It is also called as a viral chromosome. Viral genomes can vary from few thousand up to 100,000 nucleotides. It means there are some viruses that have genome or that have a chromosome that consists of few 100,000, and there are some viruses whose genome can be up to 100,000 nucleotides. Viruses with RNA genome. Almost all plant viruses and some bacterial and animal viruses are with RNA genomes. Genomes are rather small in case of those viruses which are with RNA genome, and it can be up to few thousand nucleotides. Viruses with DNA genomes. They often have a circular genome. Like in case of lambda virus, the size of the genome is 48,000 base pair, approximately 48,000 base pair. Replicated form of viral genomes. All single-stranded RNA viruses, they produce double-stranded RNA molecules, double-stranded RNA molecules or DNA. Many linear DNA molecules become circular. Viruses and kingdoms. Many plant viruses contain single-stranded RNA genome. Many fungal viruses, they contain double-stranded RNA genomes. Many bacterial viruses contain double-stranded DNA genomes. Here we can see that genomes in the viruses that can be in the form of DNA viruses. We can classify them as DNA viruses, RNA viruses, RNA DNA viruses. So DNA viruses, they can be single-stranded DNA, they can be double-stranded DNA. In case of RNA viruses, again it can be single-stranded, double-stranded, while in case of RNA DNA viruses, that can be single-stranded RNA and double-stranded DNA viruses. Here we can do a comparison between the viruses and the number of genes which are present on the genome of those viruses. The example is of parvovirus. The host are the mammals. The type of the genome is single-stranded DNA, and the number of genes which are present in parvovirus, they are 5. Like in case of lambda, we can see here lambda, it is a double-stranded DNA virus and the number of genes are 36. Another example can be tobacco mosaic virus. It can be single-stranded RNA and the number of genes are 6. Influenza virus, single-stranded RNA genome and the number of genes which are present on the genome of influenza virus are 12. So this is a comparison of the genomes of the viruses and the number of genes which are present on the genomes. This is another example of the comparison. We can see here adenovirus and the number of genes 30, hepatitis B virus, the number of genes, then influenza virus, parvovirus, and the, let's say at the end, tobacco mosaic virus, approximate number of genome size 6.4 kb, and the number of genes which are present on the genomes, they are 6. So this is a comparison between the size of the genome of some of the viruses and the number of genes which are present on the genome of those viruses. We can see that genomes in the viruses, they can be in the form of single-stranded DNA, single-stranded DNA positive strand, single-stranded DNA negative strand. It can be in the form of single-stranded RNA. It can be in the form of double-stranded RNA. It can be in the form of single-stranded RNA virus, single-stranded RNA virus with a positive strand. This is single-stranded RNA with negative strand, as well as double-stranded DNA virus. The example is T4 vaccinia. So, in case of viral genomes, we can see that genomes are in the form of DNA as RNA. If there is DNA, DNA can be single-stranded, DNA can be double-stranded. And if the viral genome is in the form of RNA, RNA can be single-stranded, let's say like positive strand. It can be in the form of negative strand. This is the example of genome of tobacco mosaic virus. The size of the genome of tobacco mosaic virus is approximately 6,400 nucleotides. There are 3 essential genes which are present on the genome of tobacco mosaic virus. One gene, this is the gene number 1, replication associated protein. Another gene which is responsible for the production of movement protein. And other gene which is producing coat proteins. So the genome of tobacco mosaic virus consists of 6,400 nucleotides, and there are 3 essential genes which are present on the genome of tobacco mosaic virus. Another example about the genome of pox virus, that is a typical large double-stranded DNA virus. The genome of pox virus consists of 180 kb DNA, and there are approximately 100 genes which are present on the genome of pox virus. The third example about the viral genomes is of the poliovirus. Poliovirus is a single-stranded positive sense RNA genome that is approximately 7,500 nucleotides long, and these are the genes which code for specific polypeptides and the proteins which are present on the genome of poliovirus. Genome of the pox virus is linear double-stranded DNA between 130 up to 375 kb. There are long hairpin structures at each terminus. The genome of pox virus encodes 150 up to 300 proteins. Coding regions are closely placed in case of genome of pox virus, means there are no introns, and the coding regions are on both strands of the genome because the genome of the pox virus is double-stranded, so the genes are present on both strands of the genomes. If we conclude about the viral genomes, that viral genomes can be single-stranded RNA genomes, it can be double-stranded RNA genomes, it can be single-stranded DNA genomes, and it can be double-stranded DNA genomes. It can be in the form of a linear structure, and it can be in the form of circular structure. So this is all about the viral genomes. So viral genomes can be linear, viral genomes can be circular. It can be in the form of DNA, it can be in the form of RNA.